This is talking about the taphonomy of cell preservation from a very neat study shown down here at the bottom. Early Tonian macroalgal fossils. Macroalgae in this case means probably they're related to plants, probably not cyanobacteria, which some people would call microalgae, although that's kind of ridiculous because they're prokaryotes. Yeah, they're bacteria, and it's in the name. Shown up here is our little inspiration for the day. Here's a little algal cell dividing, a little microbial or microscopic photosynthetic cell. There we go. So, shown here are some really fantastic fossils, and there are several very nice pictures. I just pulled one of them from this paper. And shown here are the elemental maps of these things. So we get a lot of silicon in here, but look, the silicon is lighting up, it's the green color, outside of the fossil. So the silicon is coming from the substrate. Same thing with the aluminum. Oh my gosh, are we in an aluminum silicon type environment? Aluminosilicate? Is that what clay is usually made of? You betcha, yeah. With a fair amount of potassium? You bet. A lot of potassium out there. Same thing, a little bit of magnesium, but it's kind of low magnesium that's making this up. But look what the cells are made of. These are iron. That's a little tiny Iron Man. So these cells are probably not originally made of iron, just because I would say most algae today are not constructed entirely of iron. Now, are they? Yeah. This is probably diagenetic change, which probably led to their preservation. So this is rapid deposition of iron on the surface of these, and we know one way that that can happen is in the form of iron sulfides like pyrite, for example. There are other ones, but that's going to be our shorthand for iron and iron sulfur compounds. Shown over here, this is a map of the carbon, which you can see some little blobs of carbon around it. These may have come from bacteria or other little organic bits that were in the same environment. You can definitely see a little trace here of carbon running up through it in this middle fossil and in the top fossil. And this bottom fossil is of something else. And so here you can see it's pretty much just the background area here. So these top two look as though they are macroalgae cell fossils, and the bottom one looks as though it is just a blob in the matrix or something else that is not a macroalgae pyritized fossil. You can see from the original image up here that here are the two little fronds that they're going to examine later. This is this one, this is this one turned over. You can see how difficult they are to see in the actual rock. You would have to cut open the rock, polish it off, and then do fluorescence microscopy or EDS or something like that in order to actually see these elements pop out and get a little signal, because it is the trace of iron that is actually marking the position of the cell in these images. And the best thing about this paper is that they actually looked not just at the exceptional fossils. So here are some of the best preserved fossils. Once you see these little rings, these little iron rings, and they've rusted a little bit, they've oxidized, so that they're a little bit sort of rusty brown color. But you can see that, yeah, those are actually picking out what appear to be the junctions between cells. So where one cell wall pushes up against another one. And it would appear that that is the site where this iron was precipitating. But nearby, they will have, they call them grade 1 for the best, grade 2 fossils, and grade 3. Now, the grade 2 fossils actually show the same sort of cross walls, and these are compressed, and these are found in the potassium-rich clays. The iron-rich clays are where you get the super exceptional grade 1 preservation, which doesn't look all that good to the naked eye, but under certain types of illumination that show you the iron specifically, they look fantastic. Ah, so good. And then you've got these grade 3 fossils from nearby, but you can see that there was some iron present, but it's really been oxidized, either at the time of fossilization or in the process of diagenesis afterward. And so, because it is so oxidized, it's no longer clustered, and you don't get those little divisions between cells showing up. You get just sort of an orangey smear that picks out at least some of these divisions between cells. And so these are all thought to be from this. They're all from the same formation. They're thought to be from different depths and slightly different environmental conditions. So if the clay had a lot of potassium, then you would end up with grade 2 type fossilization at this one site at this one time. But maybe we can extrapolate these things to other sites at other times. That's always the goal with science, to get some generally applicable knowledge. We can see that we have fantastic fossilization, although everything is flattened, but they were only as thick as a cell to begin with, so, not that much of a change. But yeah, that's where we have the best preservation there, including some of these little sort of lines running down the side of it. And rather poor, but still cellular level preservation here, where there was a little more oxygen present. We have poorly pyritized cell boundaries. So there you go. What aspects of their original biology do these fossils retain? Nothing that they showed in there. There's a little bit of carbon marking the cell walls. So presumably that's coming from something like cellulose and all the other extracellular matrix proteins, and maybe also the membrane, which those would all be in there together. And they don't appear to be resolved. Like, you can't tell the cell wall from the cell membrane in these fossils. They're exceptional. It's just that you got to be, the exception to exceptional to get that kind of subcellular preservation. What aspects have been lost? That's the same question, and it's most everything. DNA, there's no nuclear fossil that they were able to locate inside of these. How did the preservational environment alter the appearance of what were probably the same macroalgae? And so that's basically this slide. Depending on what was in the clay and what conditions were around at the time, they get different deposition and alteration of mostly metal ions that kind of determine which features are left of the fossils that we see. And the last one is, what's a debrite? A debrite is formed from debris, like here's stromatolites that are breaking and macroalgae that are breaking and falling down the hill into the anoxic zone and are becoming fossilized there. They're getting covered over with a little bit of mud, either gradually or in an event like a storm, something like that. And are ending up being the lucky ones that get preserved, basically forever. Yeah, for a couple 100 million years anyway. So the time, the Tonian, is actually the time. So there's the Ediacaran at the top of the Neoproterozoic, and then there's the Cryogenian in the middle, where we think the Earth froze over. We'll get to that. And then there's the Tonian, which is at the bottom. It's about 280 million years, if I remember right. And the earliest probable sponge fossils come from the Tonian. They're actually little reefs of some sort of organism. Appears to be cell-based, appears to be water filtering. And it's so far back in evolutionary history that we probably shouldn't think of this as a sponge. This is just what, some eukaryotes looked like that were around at the time. And they are like a crown group to the crown groups that will form the Ediacaran biota and then form the Cambrian biota, which then go on to be replaced by slightly different sort of Mesozoic and Cenozoic biota, such as ourselves. Yeah, good for us. Yeah, there we go. So yeah, there you go. I think that's good enough for this one.